Now we move on to the next part of the chapter, which is the non-Mendelian inheritance. In this, the first topic we will see is incomplete dominance. In this, incomplete dominance is the inheritance in which the offspring show characteristics between two extreme parental characteristics. Now, in this, the progeny or the offspring which is produced does not show fully dominant character or fully recessive character. It shows the characters between them. For example, we take the flower color in Snapdragon. Here, the red is and white flower will give a pink color flower, which is an incomplete dominance. Let's see how does the crossing takes place. Okay, so here we have capital R, which is the red flower. We have small r, which is the white flower. This capital R and capital R is the dominant character, while small r is the recessive character. These are the parents. Now the gametes we take is capital R, small r. We pollinate this and the F1 generation we get is capital R and small r, which is pink color flower. This is the incomplete dominance. This contains half capital R and half small r characters. Now, using a Punnett square, we will try to get the F2 generation, which is capital R, capital R, capital R, small r, red, pink, small r, capital R, pink, small r, white. Now we go on to the ratios, which is the first one is phenotypic ratio. 1:2:1. How is this possible? We got one red flower, two pink flower, and one white flower. So the phenotypic ratio is 1:2:1. One red flower, two pink flower, and one white flower. Similarly, the genotypic ratio is same as the phenotypic one, which is 1:2:1. Same thing, 1 capital R, 2 capital R, 1 small r. The second non-Mendelian inheritance is the codominance. Codominance is the inheritance in which both allele of genes are expressed in hybrid. Example, ABO blood group in humans. What is codominance? Codominance is both the allele of the genes are expressed in the progeny. Example is given is ABO blood group in humans. Let's see how. ABO blood group are controlled by gene I. Gene I has three alleles, IA, IB and small I. Here IA and IB are codominant alleles and these are dominant over small I. IA and IB are dominant characters while small I is the recessive one. It controls the production of sugar polymer from plasma membrane of RBCs. The gene I controls the production of sugar polymer from plasma membrane of RBCs. IA and IB produce a sugar which is slightly different from small I. Let's see the blood group and their genotype. A blood group has a genotype of IA. Blood group B has a genotype of IB. AB blood group has a genotype of IA and IB, and O blood group has a genotype of small I. The third one Non-Mendelian inheritance is multiple allelism. It is multiple allelism is a condition in which more than two alleles govern the same character. This is the same thing which we have seen over here on the top. Over here, IA and IA are codominant, where both the codominant are dominant over this recessive small I. So here the example they have given is alleles of ABO blood group. Three alleles are present, IA and IB, and small I, from which IA and IB are dominant over small I. Now, the next topic is polygenic inheritance. Now, what is polygenic inheritance? It is the inheritance in which traits are controlled by three or more genes. Before, we saw that the characters are two, dominant and recessive, like tall, dwarf, round, yellow. Over here, polygenic inheritance is controlled by three or more genes. For example, human skin color and human height. Human skin color cannot be distinguished upon two colors. It has many colors, while the human height does not only have two specific height but various heights. Now, polygenic inheritance also takes into account the influence of environment. In a polygenic trait, phenotype reflects the contribution of each allele. What we see reflects what kind of allele have given the contribution. Now, the example one we see is human skin color. Now, human skin color has three types of polygenes in them: A, B, and C. All three of them are capital A, capital B, and capital C, which you can assume or we can tell as these are the dominant one. So, what will be the recessive one? Small a, small b, and small c. Okay, now let's go to the other thing. Genotype of dominant alleles. Genotype of dominant alleles are capital A, capital A, capital B, capital B, capital C, capital C. Now these, when it comes, shows us the darkest skin color or the dark skin color. Genotype of recessive alleles. Genotype of recessive alleles is small a, small b, small b, small c. When these are expressed, we see light skin color. Now, we not only have dark skin color and light skin color, we also have a intermediate skin color. So the genotype of the intermediate skin color is capital A, capital B, small b, capital C, small c. The next one is pleiotropy. Now, pleiotropy is a phenomena in which a single gene product may produce multiple or more than one phenotypic effect. I repeat, pleiotropy is a phenomena in which a single gene or a single factor product may produce multiple or more than one phenotypic effect. So, the single gene can show us or give us multiple phenotypic effect. What is phenotypic? The things which are seen through our eyes physically are phenotype. So, a single gene can show us many phenotypic effects. Now, for example, we have starch synthesis in pea plants. Other is phenylketonuria and sickle cell anemia. Now, phenylketonuria and sickle cell anemia are genetic disorders, are genetic diseases in which the mutant gene exhibits many phenotypes. Here, the gene will exhibit many phenotypes. Now, due to this, the disease phenylketonuria results in mental retardation, while reduction in hair and skin pigmentation. The next example is starch synthesis in a pea plant. Starch synthesis effectively by capital B gene. Therefore, large starch grains are produced. Now, due to capital B genes, large starch grains are produced, while due to small b genes, we have less efficiency of starch synthesis, and small b produce small starch grains. Now, if we see the crossing between the capital B and small b, what we get is, first we see the gametes, that is the capital B, small b, then we cross them and we get capital B, small b, which is the intermediate sized seed. When we cross it, we get intermediate sized seed. Capital B has large seed size and small b has small seed size, which together gives us intermediate sized seed.